Hey everyone, so I'm going to quickly introduce Dr. Prabhu, who is a seasoned professional with us at Care Day, has been here a couple times, and has made some great strides in the field relating to audiology. So Dr. Prashanth Prabhu is an assistant professor in audiology at the All India Institute of Speech and Hearing, Mysore. He holds a PhD in audiology from IISH and works in the area of auditory neuroscience with a focus on misophonia, tinnitus, and hyperacusis. He has authored over 80 peer-reviewed publications and contributed to 8 book chapters. Dr. Prabhu is a member of the Misophonia Research Network. He also serves in editorial roles for Frontiers in Audiology and Otology, Post One, Hearing Balance and Communication, and the Egyptian Journal of Otolaryngology. Today, he is presenting findings from his recent publication, Auditory Efferent Pathway Functioning in Individuals with Misophonia. Just a reminder before we ask all speakers to try their best to not make any triggering noises during the presentation. And I'm going to give it away to Dr. Prabhu.
Thank you, Grace, for the kind introduction, and thank you for the entire team for inviting me to talk on one of our publications, which talks about auditory mechanisms of misophonia. I'll try my best to not produce any triggers, and if something happens, without my knowledge, I apologize in advance. Okay, so what I am talking today is about a study that we conducted, which talks about the auditory mechanisms of misophonia. So in our lab, we have been studying misophonia from an audiology point of view or from a hearing perspective, mainly from a neuroscience background, how the processing happens at the level of the auditory system. Maybe the processing itself is affected, so that might be causing misophonia. So that was the main hypothesis under which we have been carrying out our research. So the agenda for today would be that I would be talking about, in brief, about how our ears function and what is this auditory efferent system. So I'll try to explain it in a simple terms, what is auditory efferent system and how did we do the study and what was the result of our study that we did. And I'll talk briefly about what are the future research that we are doing in our lab to understand misophonia better. So all of us know that we have our 5 senses, and hearing is also one of the important sense. And hearing has 2 functions. So one is the hearing from birth till the geriatrics as well, and also it's important for balance. Okay, so hearing has mainly 2 major functions, that is hearing and balance. So let us try to understand how we hear. Just in brief, there would be sounds that we are hearing. So the sound waves would pass through our outer ear. We call it as the pinna, and it goes and reaches something called as our eardrum or the tympanic membrane. So which is a thin membrane which vibrates, and it vibrates the 3 bones that you are seeing here. Okay, so these are the smallest bones in our ears. So these would vibrate, and through this is called our middle ear. From the middle ear, it goes to the inner ear, which is the snail-like structure that we see, and that is connected to the auditory nerve through which the sound information is sent to our brain. So this is, in brief, how we hear. It starts from the outer ear and ultimately it reaches our brain. So what are we discussing today is about an article that we published in 2024 on auditory efferent pathway functioning in individuals with misophonia, and it's a free access, and it is listed also in the soQuiet website. You can download and read it in detail if you're interested. So I'll try to quickly summarize what we did. So I said that from the ear, the information will go to the brain. Okay, so that's the normal pathway that happens. So and we have something called as 2 pathways. So the pathway that I just said is from the ear, the sound will reach to the brain. That is your afferent pathway. That is, it starts from the letter A, afferent, that is from the periphery, that is from the ear to the brain. But there is also something called as an efferent pathway. So what is the efferent is, there would be information that will be sent back from the brain to the inner ear. So remember, this is a snail-like structure, that is your inner ear. The functioning of the inner ear gets controlled by this efferent auditory pathway. So how this functions will be controlled by the information that comes back as a loop through the auditory system called as the efferent auditory pathway. So what is the function of this efferent auditory pathway? So to make it clear, our brain helps to our ears to handle the sounds. Okay, so if we did not have the efferent pathway, what would have happened is all the sound information would have directly reached the brain, and it would have been too much. So to how much sound the brain will actually receive and process would be controlled by this efferent auditory system. So to give an analogy, your efferent system can be considered as something in the brain where there's a sound engineer who would control how much of the sound will be actually heard by the audience. So let's imagine the microphone to be your ears, ears to be your microphone, and the brain as a sound engineer in a recording studio. Okay, so what is the output would be decided by the sound engineer. So the microphones will pick up all the sounds, but the sound engineer or the brain would control how much information will go in, because otherwise it would be too loud or chaotic. So the auditory system is like an engineer turning down or balancing the volume. So that's the function of your auditory efferent system. So it's basically a two-way street where ears send the information to the brain, but the brain also sends the information back to control the way that the ear is receiving the sound, and it's ultimately getting processed in the brain. So basically there is a forward pathway and there is also a backward feedback loop that actually occurs. So it's a feedback pathway, that is signals go not only from the ear to the brain, but also back from the brain to the ear. So what is its purpose? So why does it happen is because for us to tune, protect, and filter the incoming sound. Okay, so when you're in a noisy environment, I've given an example where the brain tries to tune out for one person whom we are talking to, and it will ignore the other people who are actually talking in the restaurant or in a noisy environment. How does that happen is because of the efferent auditory system. What does it do? It will reduce the output of the surrounding noise, and it will increase the volume of the sound that you actually want to hear. So it will basically do something called as tuning. Just like the sound engineer, you want the high frequency to be boosted, or you want the speaker to be enhanced, but you want the noise to be reduced. You want to reduce the low frequency. So that's how your efferent auditory system will control the activity of your ears. So it's our built-in volume control. Basically, efferent system is your built-in volume control, so that will try to control the information that ultimately reaches the brain and it gets processed. So to give another naive example, it's like if you use your sunglasses for your eyes because you don't want the entire sunlight to go in, you want it to be protected. Just like that, efferent system is also like the control mechanism where it tries to protect the sounds from getting harmful sounds or harmful input to the brain, basically. So it protects from loud sounds like automatic sunglasses for the ears. It helps to focus like a party where we tune into only one voice. So if you're anxious, what can happen is it may not filter well. I mean, if it may not properly function, making even the soft sounds feel, very intrusive. So this is the major function of efferent auditory system. So we basically wanted to check whether is this efferent auditory system or the efferent functioning is affected in individuals with misophonia or not. So we hypothesized that whether is this volume control works differently in those with individuals with misophonia. So we just wanted to check maybe the control in the sound box of the sound engineer or the might get struck. Okay, so it might be not working properly. So because of that, the sounds or the trigger sounds, which was supposed to be controlled by the efferent auditory system, it may not get controlled, and that might be passing to the brain, and that might be leading to misophonia was the hypothesis that we had. So the volume control and the filtering may not work in the same way was the hypothesis. So maybe the system doesn't turn down the certain sounds, like having a super sensitive microphone that picks up even the smallest sounds, and it is not actually affecting it. Okay, so these isn't a weakness, but a different way in which the ear-brain loop processes sound. So this efferent system abnormality is seen in several auditory conditions. We have it in hyperacusis. This is affected in individuals with auditory processing disorder, etc. So keeping that in mind, we wanted to check how does this function in individuals with misophonia. So how did we do that? We did using a phenomena or a mechanism that we call it as a contralateral suppression of autoacoustic emissions. And it might be too technical. I'll try to break it down for you. So as we know, the sound goes to the inner ear, and there is a structure called as cochlea, or a snail-like structure, which has something called the hair cell. Okay, this is how it moves. I mean, there are hair cells. There are like 13,000 hair cells inside our inner ear, which will move like this. Okay, so how much movement happens would be controlled by the efferent fibers, or your efferent fibers will control how much movement would happen. If your efferent system is abnormal, what happens is there would be extra movement, or there will be uncontrolled movement, which leads to additional generation of something called as autoacoustic emissions. What are autoacoustic emissions is there will be some emissions or some sounds that will get generated from the cochlea. So from your inner ear, there will be sounds that get generated. How much sound would get generated would depend on whether your efferent system is normal or abnormal. So just to give it example, let's consider the cochlea as a tap and the water that is dripping to be the autoacoustic emissions. So you have your tap, which is your efferent auditory system, which would control the amount of emissions that will actually come. But if your knob of the tap is abnormal, what happens is this becomes uncontrolled. You are not able to control this. There would be extra emissions that would actually get generated. So we wanted to check whether are these extra emissions getting generated, or efferent system is not able to control. That might be leading to the triggers or the abnormal auditory perception that might be happening in individuals with misophonia. So we wanted to check that. So what the major objective of the study was to evaluate the efferent auditory system functioning in individuals with misophonia, with and without misophonia, using contralateral suppression of some transient evoked autoacoustic emissions. So what did we do?
Dr. Prabhu, I don't think your slides are advancing. We're still looking at the first slides. We're looking at your PowerPoint window, and we're still on the first slide. So if you've advanced, we're still looking at the first. I just want to let you know we're not seeing the slides as you're advancing.
Is it okay?
Okay, there we go. We're on methods now.
Okay.
So I didn't want to interrupt, but people are like, we can't see the slides. Okay, thank you. That's good.
Can you see it now?
Yep, we're looking at the one that's titled methods. We were just on the first slide. We hadn't seen them advance until now. I'm just letting you know.
Oh,
okay, keep going.
For that.
You're okay.
Yes. Yeah, okay. So I'm sorry for that. So, okay, so in the methods, just to quickly tell you that like 30 controls, 30 individuals with misophonia. So 15 had mild and 15 had moderately severe. And we did not have individuals, we ruled out individuals who had tinnitus or hyperacusis. And what we actually found was there was no significant difference between the two groups. So even in both mild and in the severe misophonic groups, we did not find an efferent auditory system abnormality. So the efferent auditory system was actually intact. It was functioning normally. So that's what we found. Okay. So the major result that we believe it is not the efferent system which is contributing for the generation of misophonia. It might be something other than the auditory efferent system. We just wanted to rule it out. This was our hypothesis, and our hypothesis was rejected. So basically, this was not the reason for generation of misophonia. So we are at our lab, we are looking at the other aspects, so what are we looking at is more at the cortical level, not at the, mainly in the afferent pathway, not in the efferent. So we are trying to look at the auditory cortex and the possible connections that are there from the auditory system to the additional areas in the brain. So just to have a quick sneak peek, so we are carrying out multi-channel auditory late latency response that we have submitted an article and it's under review, and where we are finding abnormal waveforms and we are finding differences in the way the auditory cortex is responding. So, for example, it is the processing in a misophonic group or the areas that get activated are different in individuals with misophonia and in the control group. And the source of generation of these potentials were also found to be different. I think once it's published, maybe we'll discuss more about it in the next case session.
Dr. Prabhu? Dr. Prabhu, this is Zach. Hey, your methods, we're still on the slide with the methods. I don't know if you could please advance the slides as you move through so we can see them. Thank you. Now we're on training the sound engineer. Thank you.
Oh, I don't know why. I think maybe when I'm going to the full screen, it is not coming.
That's probably it.
Anyways, I'm not sure. Yeah. So this is the research that we are doing. That's what I said. So we are finding differences in the waveforms, and we are finding differences in the way that the brain is responding or the brain areas that gets activated. So the article is still under reviews, and hopefully we'll be publishing it, and I'll be able to share these results in the next session maybe. And so the whole idea is the efferent system isn't broken. I mean, that is not what it is. So basically we need to train the engineer to learn better control. So I think the slides weren't visible. I just quickly show you. So I started off with this. So, and then we learned, this is what I wanted to show with respect to how we hear, the basics of it quickly for a minute. And this is the article. You can, it is already available. You can download and quickly see it. So I guess, so the major thing is what I wanted to tell is with respect to the tab. So we control the autoacoustic emissions through the efferent auditory system, and that is what we did in our study. Okay, and the result, we found no significant difference. So thank you for having me. So I'll be happy to discuss and take any questions. And I apologize for the slides that did not move. So, we had checked and double checked. It was
that's okay.
Fine. Yeah. So, anyways,
it's live theater. It happens. Thank you for being patient.
Okay.
All right, we have some questions.
Thank you, Dr. Prabhu, for your presentation and for adjusting on the fly with problem solving. We appreciate that flexibility. I'm going to just tee up a couple questions for you, if that's okay. We've got 5 minutes for some good conversations.
Yes.
So the first question here I have, where did it go? It just went away. Excuse me. Our first question here is, can you describe how tinnitus and misophonia are related, if they are related at all?
Yeah, so tinnitus and misophonia, so what we have found is, so there is a small percentage of individuals who might have tinnitus associated with misophonia, but not everybody with misophonia will have tinnitus. So we can say around 15 to 20% of individuals with tinnitus have misophonia as well. So the way that we can explain the relationship is with respect to, the Prof. Pavel Jastrubov, they explain it with respect to the neurophysiological model, where there is an abnormal linkage between the auditory system and the limbic system and the autonomic nervous system. So that is affected, might be similarly in individuals with both tinnitus and misophonia. So considering that, they might get related, but there is, tinnitus can be a comorbidity, but not everybody has it. So, and the relationship is the similar network that might be there, that might be leading to misophonia is what is known as of now.
Excellent, thank you. The next question here for you is, do you recommend seeing an audiologist to see if you have misophonia or another auditory processing disorder?
Yeah, so an audiologist as such, wouldn't diagnose misophonia as such, because it is always a teamwork where we can understand it in a much better way. But how would I recommend is when we go to an audiologist, so an audiologist would rule out other comorbid conditions, like we can rule out tinnitus, and most importantly, we need to rule out hyperacusis, which is again a comorbid condition with misophonia. So ruling out that would be done by an audiologist. And auditory processing problems. So we are finding auditory processing issues in our studies where we are doing, but we are yet to establish maybe a gold standard or a auditory test battery to identify misophonia yet. So until we come up with that, right now it's basically ruling out other comorbid disorders rather than diagnosing misophonia.
Thank you. I'm going to combine two questions here into one for you because they're so similar. So this is one question brought together from two different questions. The question is essentially about other experiences when triggered. So one person asks about, is painful or tightening inside the ears when exposed to trigger sounds, is that common? And another person asks about dizziness in response to certain sounds, whether that is part of misophonia or maybe something different. So can you comment on dizziness or inside your ears, a sense of pain or tightening when triggered in misophonia?
Okay, so the way that I would try to explain is whenever somebody has misophonia, so there would be a fight or a flight response, or there would be activation of the autonomic nervous system, which can lead to several bodily sensations. So mainly there might be tightness in your arms or your legs, and there might be any bodily sensation that can occur. So considering that, what can happen is the tightness or sometimes the pain that you can feel, or maybe a fullness in the ear that you might feel. So all that might be related to the anxiousness, or basically the activation of the autonomic nervous system, rather than an auditory physiology point of view. And even from a giddiness perspective, there is no direct relationship that we can explain with respect to giddiness associated with misophonia. So, but there is one angle that we are also trying to look at is migraine is something that is common in individuals with misophonia as well. So we are looking into that aspect as well. And there is a common entity, we call it as a vestibular migraine, where migraine is associated with giddiness. And there are individuals where we have surveyed, where we have tried to find out the presence of vestibular migraine. We are finding that many individuals do have vestibular migraine. So we are trying to explore on that front as well. Do they have any vestibular issues that might be leading to the giddiness as such?
Okay, thank you for that. I know we're right at time, but I'm going to try to squeeze in one last very clinical question for you from another clinician. And maybe you could try to answer this in 10 seconds or less. I'll put you on the spot, Dr. Prabhu. So this is from a clinician. It's a specific question about auditory processing. Mm-hmm. Can you comment on whether we should be calling for auditory processing evaluations, which is in the US different than an audiology evaluation? So for misophonia, when being treated by an audiologist, should there be an auditory processing evaluation that's done?
So my quick answer would be yes. As of now, based on our studies, we feel that let us do some central auditory processing, mainly the cortical processes like the binaural integration and temporal processing assessment. So that might be affected. So along with the traditional audiological tests, assessing central auditory processing is something that we can add on in our audiological test batteries.
Thank you for that. And thank you for the quick and very definitive yes answer to that. Heidi, I hope that is helpful for you in the work you do and for any of the audiologists who are here in the work they do. We're going to be here at the end. We're going to shift over now to, we are at the end, I should say, and we're about to shift over to our next presentation with Dr. Heller. A question I just want to ask you real quick, Dr. Prabhu, one person asked about whether you would make your slides available for us to share with people, and I wonder if that's something you're open to.
Yes, I'm open for that. So because the slides also didn't move as well, so I think it's better you can share it with the participants. I'm fine with sharing.
Excellent. So for that individual and anybody, you can reach out to us directly if you would like the slides. Also, we'll maybe look to make them just available for download on our website when we post the recorded videos after Care Day. Thanks, everybody. We'll see you in the next session.
Yeah. So thank you for having me.